This is a production of Cornell University.
All right. So we've talked about, we just finished this lecture. We talked about why we look at things, okay, and the classification based on what we're interested in. So what I'd like to do today is think about what they're doing on the landscape, the effect of soil organisms on the environment. Okay. Now, at this point, we've done this safari. You've got like everything from nitrogen fixation to, structural type of development, things like that. But I'd like to start taking a look at two sort of very interesting groups of organisms that are in the soil and sort of follow what they do. Okay. Now, I start with this slide. I'm sure a lot of you have seen this this is the classic sort of soil science slide where you have a hand holding some soil. But I want us to start this lecture by thinking about what's going on in this. Okay, this is the volume of soil. Okay, now it could be in the soil landscape someplace. It could be in a pot. It could be, but what's going on in here? Now, Carl Sagan never said this, but he's often attributed to saying, there's billions and billions of stars in the galaxy. He never actually said that, but we often say, there are billions and billions of microorganisms living in a grain of sand or in a teaspoon of soil. Have you guys ever heard that? There's a multitude of organisms just in a teaspoon of soil. And the reality is there's more organisms standing underneath you than there is above you, right? Does that make sense? Okay. Well, let's actually talk about what's going on in here and what are the organisms that are in this space and how they have an effect on this volume. Now, usually when we think about this is the picture that we come up with. Okay, we sort of, it's the classic trophic graph. Okay, where we're looking at primary producers, primary consumers, and so on, all the way up to the detritivores. Okay, and then starting the cycle again. Okay, but I'd like you guys to think about it in another sense. I'd like you to think about it in a sense of form, yes, certainly form and function, which you sort of see here. But I also want you to think about it in a sense of diversity and what that diversity is creating. Now, you've seen a slide that's very similar to this earlier, okay? And when we think about diversity, we talk about, this is size, genetics, energy, and trophic level. Did we just talk about that? Basically, the four ways that we classify things, okay? But I want you to think about this diversity in the sense of what are these organisms actually doing? Okay, yes, we're interested in them and we're going to classify them that way, and that's why we classify them that way. But what are they actually doing? And to think about what they're actually doing, let's think about where they are and their numbers and their forms and all the things that we've basically just talked about in the last lecture. Okay, so biomass. Okay, here's that mushroom that's sitting out in Oregon someplace. It's not a mushroom, it's a fungi. Okay? The number. This is one organism. We have earthworms. We have all the way down to bacteria. An abundance of these guys. Where they are. Okay? Dry versus wet. Cold versus hot. And what they actually do in that system. Their functions within that system. So let's think of them as a functional relationship. What's going on functionally? Okay, so these soil organisms play these critical roles. What are these functions? Okay, it turns out these functions are basically, they can be beneficial, they can be neutral, and they can be detrimental. And what I'm going to do is we're going to look at a model system. The model system we're going to be looking at is earthworms. And we're basically going to be looking at three things that this earthworm does, or earthworms do. Issues of soil structure, issues of infiltration and aeration, and then finally, carbon and nutrient cycling. Okay, so let's start with an earthworm. There's our earthworm. Okay. This is basically a Wikipedia pull. I just pulled it straight from Wikipedia. What is an earthworm? Well, it's a tube-shaped, segmented animal that is commonly found living in soil. Its digestive system runs straight through its body, and it conducts respiration through cuticles covering its skin. It has a simple closed blood cell circulation system. They're hermaphroditic. They are as an invertebrate, it lacks a skeleton, but earthworms maintain its structure with basically fluid-filled systems. These fluid-filled systems basically help with mobility. Okay? Basically, this is a Wikipedia pull. This is what you would get if you pulled on Wikipedia. Okay? So let's take a look at soil structure, infiltration and aeration, and nutrient cycling. What is this worm actually doing? Okay, let's start with soil structure. Okay, generally when we think of soil structure, can you guys see this slide? Yeah? Generally when we think of soil structure, this is what we think of when we talk about worms. The worm casts. You know, it comes out in a rain, or you go out to the soil, the field, and you can see these sort of casts coming out of the soil. Okay? This is the detritus coming out of the tail end of the worm. Okay? It's not poop like we would think of it as poop. Turns out that worms are gut, basically they are gut grinders, they're gut fermenters. Okay? And they have to have rocks and things like that in their stomach to help grind things up. Okay? They eat the material, they go through their sink, they're basically one large stomach, which is in essence what we are in a sense. One large stomach, okay? And this stuff comes out the tail end. Now, interestingly enough, this is a lot more stable structurally than the surrounding soil. Now, this is a piece of wood that somebody put the cast on, but now, if we're going to be thinking about this issue of where these casts are, we have to think about the number of worms. Okay, now this is a slide that this is actually the data that I pulled from online. And basically what this is we're looking at earthworm densities, not the casts, the density of earthworms in a silty clay loam field, basically in Indiana. Okay, and they've been looking at a variety of different field treatments for at least 10 years. Okay, they've got continuous corn plowed versus no plowed, soybean plowed versus no-till, bluegrass clover, dairy pasture with manure, and a dairy pasture manure with heavy application. And look at the distribution of organisms, or I should say the number of these organisms. Okay, plowed versus no plow, plowed versus no-till, plow versus no-till, corn versus soybean, grass system, a manure system, and a lot of food coming into that manure system. So if you just look at this slide, what is it telling you? They like manure, and they also like the no-till. Okay? But even in the no-till system, the earthworms here in the bluegrass clover mix, they're certainly higher than this. So there's a lot of things going on that affect their distribution. Certainly feedstock, certainly disturbance, certainly the type of feedstock and the amount of feedstock. Make sense? So, with that type of disturbance, these types of disturbances in that kind of population, what do you think is going to be happening with casts? Where are we going to have more casts? Pretty straightforward. We're basically looking at a trend from least casts to most casts. Now, an interesting experiment, Charles Darwin, he was very interested in earthworms. Okay? He did an experiment where he basically spread chalk over his backyard. Okay? And then he went away for 30 years and came back. Actually, he did a lot of other things in that 30 years. He came back, and then he dug in, and 29 years later, he dug into the soil to basically find where that chalk line was. The chalk line was 6 inches underground, 6 inches below the surface. Now, he spread the chalk at the surface. What had happened? Now, we don't have soil genesis this fast. And even if we did have soil genesis this fast, we wouldn't be seeing the chalk line going this way. So what was going on here? This is all earthworm casts. In 30 years, he had developed 6 inches of earthworm casts. That's where the chalk line went. Okay? Just to give you an idea about, here's that abundance. Here's that abundance. Now, do you think I would be getting 6 inches of earthworm casts here? Potentially here, though. Okay, so we got the casts. Now let's think about what other things the earthworms do. They're certainly boring lots and lots of holes. Now this is a simulated earthworm. Can you guys see the lines here? Jean-Marc, can you kill another bank of lights? Now, this is a simulated earthworm track. This is basically 4 earthworms. Can you guys see it now? Verizon, can you hear it now? No, can you see it now, sort of-ish? Okay. What you're looking at here is basically 4 simulated earthworm tracks. 4 earthworms. Now, what do you think would have happened if we were looking at that heavily manured field? If this is just 4, they were looking at 1,300 earthworms. What's that going to do to the aeration and porosity of these soils? Dramatic increase. I mean, pretty straightforward. It's a dramatic increase. So if we have that dramatic increase, what is it going to do to aeration and infiltration? A lot faster infiltration rates, and aeration is going to be a lot better. Okay? What's the effect of that on nutrient cycling and everything else that's going on in these soils? Right? We're moving materials. We're increasing materials coming in or increasing the rates of materials. We're also oxygenating the system. If we're oxygenating the system, we're increasing decomposition rates. We're basically increasing respiration rates. So things are going to be a lot more driven or faster. All right. This is an experiment where they're basically looking at macropore abundance. Okay? And what they're looking at is large versus small macropores. Okay? And what you're looking at here is earthworm populations. This is an ambient condition. They basically left the population alone. This population, they basically reduced it. In this population, they basically added more worms. And what do you see? All across, you basically see an increase, dramatic increase in the macropores, smaller increase in the micropores. So, what's going on? Yes, more population, you get more pores. But what's driving that besides the number of organisms? Here's another experiment where we're looking at cumulative infiltration rate in 3 hours. And we're looking at the types of residue present, none versus soybeans. And we're looking at basically 3 different treatments of worms. None, 15 earthworms, and 30 earthworms. Okay? If there was no earthworms, okay, the lighter color is the none. Okay, so you would think this is actually sort of inverted, how most people would do it. This is none, this is 15, this is 30. This is none, this is 15, this is 30. Okay, when there is no food present in the system, are the earthworms doing anything? They're doing a little movement, but once you start seeing food in the system, the soybeans, dramatic increases in their movement, and based on their abundance, dramatic increases in the aeration, the infiltration rate. Does that make sense? Functional response to what these earthworms are doing. Functional response in the soil to what these earthworms are doing in their own functioning. All right, let's take a look again at these casts, though. Okay, so we've looked at the stability, the structural stability, or I should say the structural creation of structure. We also looked at the aeration. But these casts do something else. Okay, these casts, we're looking at a table here that's basically comparative characteristics of earthworm casts versus soil. Okay, these are soils that are in Nigeria, and basically what they're doing is they're looking at the casts themselves versus the soils. Okay, and they're looking at a number of different things. Okay. Silt and clay percentage. If you look at the silt and clay percentage, the earthworm cast, it increases. Okay, actually, let me sit on this side. The bulk density, not that much difference, but it is a little bit less than the native soil. Okay, interestingly enough here, structural stability. This is a really cool piece of information here. Okay, this number basically represents how many raindrops it takes to destroy that aggregate. Okay, so here, number of raindrops required to destroy the structural aggregate. The earthworm cast takes basically 850 raindrops to break it up. This one basically takes 65. Now, what does that tell you about the resilience of a soil to resist raindrop destruction with the presence of earthworms, without the presence of earthworms? With, you certainly have more infiltration, things like that, but with, these aggregates tend to stay together. And we already know the benefits of these aggregates, let alone the benefits that we're going to talk about in a few moments. Okay, cation exchange capacity. We'll talk about nutrients in a second, but look at this. 14 basically to 3.5. The exchangeable calcium, exchangeable potassium, soluble phosphorus, and total nitrogen, hold off on those, but take a look and notice that in each case we're looking at more nutrients in the cast than in the native soil. Now, does that make you start thinking about what's going on with these casts? Let's take a look at this slide. This is basically a nutrient number, okay? This is basically a nutrient response to these different soils, or it's showing you the amount of nutrients that are in the cast versus the surface of the soil versus at depth. Now, I started the last lecture talking about plants and the rhizosphere. And the point of that slide was to say, okay, well, we're looking at a system where the plants are supplying stuff to the bacteria, and the bacteria in turn are supplying things back to the plants. Well, here's another relationship, or another relationship that's very similar. Because look at what the earthworms are doing. If they are concentrating nutrients in these casts, where do you think the rest of the organisms in this soil system are going to be concentrating? In the casts. So basically, these are serving as sort of host environments. Now, we're talking about microbial populations. These casts are not very big, but microbes are pretty small as well. So where is a lot of the nutrient cycling occurring? These basically are acting as fertilizer pellets. So we look at total nitrogen. I mean, if you look at basically any one of these, we're seeing basically an increase in nutrient concentration. Okay? Even when we're looking at CN ratios, there's going to be more carbon here and nitrogen, but we're looking at a CN ratio. This is perhaps the only one that, if we look at these, this is perhaps the only one that perhaps could be considered detrimental. But look at the CN ratios. They're well below 20:1. Available phosphorus, calcium, magnesium, potassium, total calcium, total magnesium, percent base saturation. And another very interesting thing here is the pH. Look at these pH numbers. The pH in the cast is around 7. The native soils, they're not that acidic. But if this cast is around 7, what is that saying to me about nutrient availability? Now, the optimum range is actually around 6.5. So we're looking at the top 6. These guys are basically where all the nutrients are, okay, and the maximum availability. Okay? Down here at below 6 inches and deeper, pH is going down. So you guys remember that curve, right? That nutrient curve? Okay, it sort of maxes out around 6.5 when we look at total nutrient availability. This is kind of really interesting. Okay, and it's indicative of something else. And let me go to this next slide. Beyond this effect of pH being around 7, okay, and the nutrient availability, it turns out that this in essence, well, I mean, if you're thinking about these casts, it helps to neutralize the acids and alkalinity of the system that may be present in the soil, therefore making root development and nutrient availability a lot better. But the other interesting thing about these casts and the tubes that they're making is they basically, as these earthworms are moving through the system, they're basically leaving behind sort of a mucilaginous type of substance that is rich in proteins on the wall of these burrows. So again, the casts and these burrows are basically acting like a rhizosphere. This one organism, in a sense, becomes a keystone organism for a lot of other organisms. Okay? This substance, in turn, serves as an energy source for the microbes, and on top of that, these casts, huge amount of nutrients, this becomes a really important ecosystem, a smaller ecosystem within a larger ecosystem. The concentration of nitrifying microbes have been observed to be about 40% higher in the burrows than in the rest of the soil. Now, these burrows, certainly if you think about it from a rooting sense, these burrows are nice large pores, nice like hole, lots of nutrients. This is also going to serve as a nice highway for roots to start expanding through. And so we're seeing what these earthworms are doing and we're seeing what their influence is on the larger soil system. It's not just them eating and surviving for themselves, but their impact on the rest of the system. Or I should say, their relationship with the rest of the system. So, questions on this? These guys are really, really cool.
This has been a production of Cornell University, on the web at cornell.edu.